Maria Paz for her introduction, and thank you very much for to the other organizers for inviting me to this astrobiology academy. I was incidentally part of an astrobiology academy myself back in 1999 at the NASA Ames Research Center. So I am pleased to give back in the same way that I've received in the past. So I will be presenting something about biosignatures of potentially anaerobic organisms in Paleoproterozoic hydrothermal vent environments. And this is work that's been long standing and that has taken various shapes. And now I'm here at the Institute for Deep Sea Science and Engineering trying to corroborate the findings with modern analogs. But I will be mostly talking about the ancient rock record. But that's the idea essentially. When we're looking at old rocks, we want to be able to compare them to relatively younger rocks so that we have a good basis for analogy. And the same goes with the biosignatures or the potential signs of life that we find in them. So I will first start by giving you a bit of the motivation behind this. And the big question behind this is, what is the origin of organic matter in hydrothermal vent environments? That is a long-standing question that also is linked to the origin of organic matter in ancient rocks. And there's usually two competing hypotheses. The first hypothesis is that the carbon is non-biological, whereby it could come from potentially the decarbonation of carbonate minerals like and you can see the chemical reaction here that's been proposed. But this usually tends to occur at temperatures that are relatively high, typically more than 450 degrees Celsius, which is significantly higher than the hottest black smokers that we know of, which are about 370 degrees Celsius or so. But there's another potential mechanism, and that's called the Fischer-Tropsch type reaction or Fischer-Tropsch synthesis, which consists in the reduction of carbon dioxide here, but this also works with carbon monoxide. It works with dihydrogen and a metal catalyst, and then some short chain alkanes from methane all the way to alkanes of various lengths can be synthesized. This is totally abiotic, and is considered to be a reaction that's likely important for the origin of life on Earth. So people like Thomas McCollum and Jeffrey Seawald have been studying this reaction for several decades. Various others have also done that. And they've investigated the carbon isotopic fractionation between the starting dissolved CO2 and the product alkanes. And they see that with the carbon number here, what's really understood is the number of carbon atoms in these alkane chains. And when the chains are, regardless of their lengths, the carbon isotopic fractionation is quite significant. We're talking about maybe around 30 per mil, 36 per mil in the cases of these experiments here. So they're quite significant fractionation that are much more fractionated actually than the typical cyanobacteria-driven CO2 fixation during oxygenic photosynthesis. So there is a way to distinguish these two, but it turns out that this overlaps a little bit the range of isotopic fractionation for methanogenesis, which is an anaerobic microbial metabolism performed by various Euryarchaeota members of the archaeal domain. And there's evidence in nature that this is occurring in the present day. So Fischer-Tropsch synthesis has been reported from the Lost City. The carbon chains were quite shorter here. A paper published by Proskorowski in 2008 in Science magazine showing fractionations of -9 to -16 per mil. And because they were short chain alkanes, they were attributed to that process. So there is indication that this is occurring today, so why not in the distant past? So we have to be careful when we're interpreting carbon isotopes or the origin of organic matter in rocks. And of course, the second hypothesis is that the carbon is biological. And here I list a number of characteristics that the organic matter should have if it's of biological origin. Like, for instance, it would be associated with fossils, or it would necessarily need to be indigenous and syngenetic to the rock, which means it needs to be in place and come from the original depositional environment. It should also be associated with some diagenetic minerals. The organic matter should also contain traces of the hydrogen, nitrogen, oxygen, phosphorus, and sulfur that's common in biomass, and also some functional groups, and have C13 depleted compositions. And here I give some examples of some nanosims images of these microfossils, which have been considered to be biogenic by Dorothy Euler and Chris House, who've separately measured trace elements and carbon isotopic compositions in these microfossils, concluding that they're most likely oxygenic phototrophic microorganisms. So here's some examples, okay? Now, of course, the motivation behind this work is to answer the question, did prebiotic chemistry or even life evolve on Mars or elsewhere? So that is, of course, the big question that us as astrobiologists are very interested in answering. And there's some hints of possible prebiotic chemistry on Mars, including carbonate outcrops and hematite concretions, opaline silica sands. These would be good places, good sediments to seek biological signatures on Mars. And recently, there's been some organic carbon, including some alkanes with 9, 10, 11, 12 carbons in the chains that were detected in soils from Gale Crater that was reported recently by Freycinet and her colleagues in PNAS. But that brings us to discuss a bit the biosignature and the terminology that we use to understand the term biosignatures and really understand what we are looking for when we say that. So I would suggest that a biosignature should be defined as a possible signature of life, because a single biosignature can hardly be a smoking gun. There's so many abiotic processes that can lead to the formation of similar biological look-alike that it's very we have to be very cautious in terms of interpreting biosignatures. And for example, as I said, there's Fischer-Tropsch type synthesis of organic carbon. There's abiotic biomorphs from chemical gardens that have been reported from various recent studies that look like microfossils. So these have to be considered as possible and plausible. Most of them are made in experiments that try to mimic also environmental conditions. And also, what was prebiotic chemistry before the origin of life, anyways? What was that looking like? It was prebiotic, so it was not life, but it looked very similar to life because it was a precursor of life. So that leads me to propose a term that is not an oxymoron, that I think is a necessity, and that is the term abiotic biosignature. The term abiotic biosignature should be defined as a biosignature that involves both abiotic processes and the decomposition of biomass. For example, you have graphite. Graphite is not a product of life, but rather of thermal metamorphism. On the other hand, the precursor to graphite, which is the organic matter, could have arisen from decomposed biomass. So a graphite is a possible abiotic signature of life. So hopefully that example makes it clear. And here in the background, I put some botryoids, botryoidal silica, that were closely associated with stromatolites. And it's been tempting for many geologists to consider these possible biological signatures. But the fact of the matter is that we don't really know the origin of these features. So I will talk about this right now because it's important to discuss these different types of biosignatures and what are the possible abiotic processes that could arise and form them. So here I'm giving six different examples of biosignatures of life in that can be seen in Precambrian sedimentary rocks. In purple, in the form of this Venn diagram, so in purple in the three upper ellipses are the molecular, elemental, and isotopic compositions of the organic matter. They're not the same. They can overlap, right? Because you can have molecular functional groups with hydrogen or oxygen and hydrogen or sulfur and hydrogen or nitrogen and hydrogen and so on. So these molecular functional groups, they go in the molecular composition. The elemental composition, that relates to the Schnapps elements that are expected to be found in ancient organic carbon. And then the isotopic composition, as I mentioned, but every one of those has a potential abiotic explanation for them. So there's a prebiotic chemical reaction I mentioned already, decarbonation, FTT synthesis. These are all things we talked about. Chemical oscillation may be new for some of you, so I will spend some time to talk about them because I think that these can lead to the formation of these kind of spheroids, these diagenetic spheroids like concretions and nodules, granules and ooids, and I will talk more in detail about this momentarily. Other sedimentological biosignatures would include stromatolites and microbial mats, as well as microfossils. But for every one of those cases, we have to consider that if we are on another planet where there's no evidence of life yet, like on Mars, and we find stromatolites, we have to consider that these stromatolites might have actually been formed abiotically and not necessarily by cyanobacterial microbial mats as we typically interpret them on Earth. So there are many caveats, and to achieve the goal of finding a signature of life, we need not only to have the most complete documentation of all these biosignatures, but also a reasonable rejection of all these kinds of abiotic interpretations. So what is life anyways? This goes to the core of the question, and it is not a trivial question. Some people say, well, we know it when we see it. Maybe that's true for a large living organism, but when we are in a microbial world, in an extreme environment, and on another planet, we really have to think carefully about this because the question of the definition of life is still not fully understood in astrobiology, and hopefully you've come across this difficulty in the field so far, and it stems from the fact that we need a lot of considerations that are both scientific and philosophical. Those problems relate to self-reproducing robots, for instance, or computer programs, even fire or viruses, or the nature of the last universal common ancestor, and the boundary between life and non-life. So I have my own working definition, which uh, is on press right now in a book, the Archean. And basically, I'm defining life as an organized and compartmentalized organic molecular system capable of self-replication, metabolism, and Darwinian evolution through natural selection and adaptation. So there's a lot of keywords in there that are important. Capable of self-replication would include viruses, but viruses are not capable of metabolism. So you see, viruses would kind of be in the middle here. They're kind of excluded from this definition. Whereas robots, like let's say transformer robots that would have organic components and would be able to self-replicate and evolve through natural selection and adaptation may be considered as living entities, even though, of course, the makers, the original makers of those possible transformers would obviously be living organic creatures. But that's another thing also. Organic, for some of you might raise some issues. Maybe you would think of silicon-based life. I think that we have to stay open-minded, and maybe we will change our ideas if we see something that could fit the other parts of the definition. But I think that carbon is so abundant in the universe and is so versatile, especially in organic chemistry, as it happens in aqueous solution, that is most likely something that we can absolutely plan to see in an extraterrestrial ecosystem or life setting. So with these definitions in hand, now we are confronting the problem of recognizing biosignatures from abiotic signatures, and that's a major challenge. And it's been a challenge in geology for quite some time, and there's a lot of objects, for instance, those ones that I list on the left-hand side and accompanied by pictures in the central part of this slide. You see a wide range of features of minerals and rocks that are often displayed in museum. Actually, a lot of these are pictures that I collected from museums. And these objects have 5 different criteria that are linking them all into what I like to call diagenetic spheroids. Diagenetic spheroids, first of all, diagenetic because all of these objects, that they be rhomboids or ooids or nodules, concretions, geodes, and botryoidal minerals, they all are suspected to have post-diagenetic origin linked to these diagenetic processes that are happening after sedimentation. They all have this circularly concentric geometry as well as radially aligned mineral patterns that makes them spheroids. So that's why I use the term diagenetic spheroids. And then they're all kind of made of the same small number of minerals, of groups of minerals, primarily silicates as chert, carbonates, phosphates, sulfides, and iron oxides. And that covers pretty much most of all the diversity of these objects. They are commonly associated with fossils and or organic matter. So they all have these characteristics, and lastly, they all have this enduring enigmatic origin. Geologists do not all agree on how they form. So that's the question ultimately that should be addressed, especially when looking for life on another planetary settings, so that we have a possible answer for at least how these objects form, which may be associated with either fossils or organic matter of biological origin. So knowing about the origin of these diagenetic spheroids could help solve many outstanding problems in geology. They are widely known to occur as ooids and granular cherts, concretions, and banded iron formations. There's rosettes and rhomboids, beautiful examples here, as well as pyrolusite occurring sometimes on botryoidal minerals, or in this case in a concretionary boulder from the deep sea in the Mariana Trench, with beautiful of these called frutexites, microscopic dendrites. So this is another primary motivation of this work. So fossiliferous concretions from the Phanerozoic, there may be a role for organic matter in the formation of these concretions. For instance, you see very often fossils in them of fish or of trilobite. This is quite common, and yet it's not really well explained by our current models of formation of concretions. So I've come across this reaction, Belousov-Jabotinsky reaction, during my time in London, and it turns out that it's a very interesting model to consider for the origin of these diagenetic spheroids. Now, these were discovered originally by this Russian scientist, Boris Belousov, and was carried forward and investigated forward by his PhD student, Jabotinsky. And it was discovered because Belousov was investigating the origin of the tricarboxylic acid cycle. So he was mixing things like malonic acid or citric acid with some oxidizers like a bromate here and sulfuric acid. You have the concentrations here and the relative amounts that I put in those Petri dishes, as well as bromide and ferroin. Ferroin is a redox sensitive dye that contains iron. So the iron goes from Fe2 plus to Fe3 plus, and that gives the different color contrast in the solution. And over time, these little spots that start as small sub-millimetric spots, they grow radially by diffusing in the solution when it's left still, and the reaction proceed, and then it oscillates by repeating another generation of a radially diffusing wing. So you have these serial rings in a two-dimensional thin film, and you can see that this is all. So these are all 10 centimeter diameter Petri dishes, and you can see the range of patterns that are made. And eventually you have those little CO2 bubbles that appear. Where's the CO2 come from? Well, obviously it comes from the malonic acid. It's the only carbon-bearing molecule that we put in here. And the malonic acid has two COOH groups, so two carboxyl groups. And this is what gets cleaved, presumably by the bromide, which attacks it. And eventually there's an electron transfer that takes place all the way to and either into the bromate or into the sulfate of sulfuric acid. And it's catalyzed ultimately by the iron redox switch that we put as ferroin. And you have spontaneous formation of beautiful patterns that are never the same, but that are always self-similar. A bit like life. Humans are never the same, they're never identical, but they're always similar. So we call ourselves human, and same thing for the other animals. So There are actually 10 patterns that can be extracted from these reactions. And that's after having done hundreds of those reactions in my laboratory and group over the years. And this is currently being written up for publication. I give them names because it's important in science to name things. And the names I gave are as agnostic as possible. I don't think they're certainly not biological. There's no biology in here to make those patterns. But some of them make stromatolitic or knobby columnar stromatolitic features. And this is part of those patterns. It's completely abiotic. The radiation, it's a radial diffusion of the rings, and you can see this at two different times. The spot proliferation here, you see this sometimes maybe on fish scales, you see this kind of pattern, or on insect exoskeletons. A circular concentricity is quite a noticeable one, and probably a telltale sign is this bilateral symmetry, or twins, as I call them, circular twins. When two circular waves meet, there's a process of destructive interference that basically creates this circular twinning, which is a kind of pattern that we definitely see in botryoids, in geodes, in concretions, and nodules, and so on. But also cavities like this is the pattern of a cavity. It's still all filled with an aqueous solution here. There are spirals. Spirals we don't see quite often in the rock record, I would say, but that's still a pattern that's formed by these reactions. And color gradients here are very intense color gradients, going from kind of a light pink here to a kind of a true deep light blue at the tip. But the color gradient is quite significant, and it also follows this concentricity. We have the color gradients in all of those, but here it's probably best displayed. So I'm proposing that what's happening in nature is that you have this oxidation of carboxylic acid from biomass using either bromate or sulfuric acid, probably catalyzed by iron or a redox disequilibrium between bromide and bromate, and that creates intermediates like bromomalonic acid or bromous acid, and eventually produces CO2, which is one of the reaction products that we see visually when running these reactions. So over the last 10 years, I've been comparing a lot of these patterns to things that are enigmatic, like I was introducing. So we have the agate geodes, circular concentricity here, beautiful cavity patterns. And you have like examples. They're not identical, but they're definitely self-similar. And it's actually probably the best explanation that you can find in the literature about the origin of these patterns, which are very well known. Anybody who has a rock collection knows about these objects. Anybody who's been in the geology or the mineralogy section of a natural history museum has seen these objects, and there's never an accompanying explanation for some reason. But they likely arise from chemically oscillating reactions. And so we've pushed this to investigate the origin of botryoids. Here's botryoidal malachite, comparing different parts of these 10 different patterns. The spiral, I can talk about this, but that's a little bit more complicated. It goes with the screw dislocation of these acicular crystals that are radially aligned in the fabric of malachite. You can see the radially aligned crystals of malachite here, and the circular concentric features. Here you have this kind of circular twin. So it's really a twin plane where the direction of the crystals that are growing there is completely changing, but kind of a mirror image of one side and the other. Um, and so I think there's definitely a good connection between the patterns, between the COR and the botryoids, but there's also further connections, including chemical connections. We've looked at botryoidal silica from the Barberton Greenstone Belt, a Paleoarchean supercrustal sequence in South Africa, as well as the Gunflint Formation in Ontario, Canada. And you can see the botryoids in plain polarized light are not quite clear, but when you cross the polarizing filter, you see the radially aligned crystals that are beautiful, forming little domains here, very photogenic things to look at. And they display not only the radial fabric, but the acicular structure and the circularly concentric geometry of these patterns. And here at the bottom of this slide is the apex chert here. You see this larger cavity, which we investigated by micro-Raman and found that these kind of puffy balls here, they are made of micron size to nanoscopic size crystals of hematite, but the hematite is also a bit ferrous. So there's a bit of Fe2 plus in the Fe2O3 composition of hematite, which is dominated by Fe3 plus, a ferric iron. So you have both ferric and ferrous iron occurring in those puffy balls inside this cavity, indicating that very much like the COR, iron, redox sensitive iron has played a role in catalyzing these reactions. More recently, we've been looking at concretions and nodules. A recent paper that appeared this past month in the journal Chemical Geology has reported these circularly concentric concretions, nodules in the Duchantuo formation. Some of them are very large. And why would they be large? Why would some be small? And then why am I even thinking about sub-millimetric size features? Well, very much like the COR patterns, I think that they display a range of size because they are fractals of the same kind of chemical reactions. So here in this case, you have a rim of pyrite, several rings, relatively circularly concentric or subspheroidally concentric rings of pyrite. Why would there be a ring of pyrite in a nodule with fossils in the middle? Well, it's a good question, but I think the answer is that the fossils represent the fuel, represent the original biomass, rich in carboxylic acid. It got oxidized during diagenesis, and radially outward diffusion of the reaction products as sulfide eventually met with ferrous iron and precipitated pyrite. And that all happened in kind of a siliceous colloidal silica precursor, presumably. And I can make a similar model for these layers that are slightly organic enriched in this apatite dolomite concretions. And these are older ones from the Paleoproterozoic. Some are meter or several meters in size. This one in the Windida Formation of Western Australia, several meters in diameter. This one from the Dashiling Formation, again, several meters in diameter. Beautiful circular concentricity, very commonly finding biomass in the form of residual organic matter or even fossils in the geometric center. For the microscopic objects, here you have some examples of pyrite marcasite rosette, again in the Duchantuo formation. Again, I'm talking more about this paper because that's a recent one. But you see here the complexity of the mineralogy is quite significant because you have both pyrite, which is a ferrous sulfide in the center, and then you have barite, which is a barium sulfate on the outside. So as a kind of a residual product was maybe diffusing outwards. And all the red in this particular hyperspectral image represents organic matter. And then you also have the carbonate in green and the apatite in turquoise, such that you have reaction products as well as reactants that are residual, that are forming these objects that would otherwise be called by sedimentologists a replacement texture. But this nucleation process, or even some people have proposed that they form from wave action, none of these processes that have been proposed so far predicts that they should have a concentric, a circularly concentric geometry, a radial geometry, or the kinds of patterns here that are seen as these twins in these various pyrite framboids. And here you can see them in, similar size, maybe slightly smaller in the rock record. We haven't done much microscopy yet on these aqueous solutions. It's quite challenging to do. But here you see two of those that are kind of fusing with one another, making twins. And here you have some marcasite, quartz marcasite rosette. Marcasite, same formula as pyrite, FeS2, and it's accompanied again with dolomite, organic matter, and apatite, forming concentric rings, which are best seen in plain polarized light here. And here in this one, you have a kind of a rhombohedral grain, but in the center, you have this kind of dumbbell shape where you see radially aligned carbonate crystals in green. in this Raman hyperspectral image, and you have a rim of organic matter around it, kind of discontinuous, but still rimming the object, and overall with a lot of little apatite grains in turquoise. So we have this model for the small objects as well as the larger objects, whereby in during earliest diagenesis we have a biological putrefaction, which is a natural phenomenon, a bit disgusting maybe for the human perspective, but still a natural phenomenon that needs to happen and that has been happening on Earth for as long as there has been life. There's been putrefaction. And then if the compounds, the reactants, the necessary reactants are there, like these oxidants, the iodate, bromate, chlorate, these have all been experimented upon by the physicists and chemists who have so far been studying chemically oscillating reactions. These can act as the reactants to oxidize the organic matter and produce this abiotic decarboxylation through chemically oscillating reactions, producing patterns which eventually will get mineralized, permineralized, that it be in silica, in apatite, in colophane, so microcrystalline apatite, or even in micritic carbonate. So this is part of the fossilization process, which involves this phenomenon of biological putrefaction, which surprisingly enough in the geoscience community, very few people are talking about. So um, these are some new ideas that I think you should keep in mind. And this is one of the latest papers I have about this, addressing this problem and comparing it to ooids. So you have here on the first row a number of different ooids that show radiation, spot proliferation, circular concentricity, all these things. These patterns are seen in COR, as I told you earlier. You see the CO2 bubbles, and then you can compare the substances. So comparing the patterns and the substances of these objects, I think, is the best way to go, because there's nothing else after that. But here, of course, we could be a lot more detailed about the substance, but we can compare the presence of organic matter, which is ubiquitous in ooids, along with malonic acid, the COR, the apatite, which probably contains some bromide, some bromine, because it's a either fluorine hydroxyl or chlorine that resides in the anionic site. There's often iron 2 plus, iron 3 plus minerals like pyrite or iron oxides in these ooids, and calcite is there and quartz is there. So you see there's a lot of connections that can be made that are based on the substances of these objects. So enough for kind of the long introduction to talk about the possible biosignatures and how to distinguish them from bona fide biosignatures. The bottom line really is that diagenetic spheroids are abiotic biosignatures that formed through COR, through chemically oscillating reactions, but they have been fueled by the presence of biomass. That is on Earth. On another planet, that could be fueled by abiotically synthesized carboxylic acid. And that's an important point, because it's not saying that diagenetic spheroids are necessarily biosignatures. They're possible signatures of life, but they've been formed abiotically. Hence why I gave those definitions earlier in my presentation. So now we'll address a problem that is a little bit more specific. I see time passing by here. That problem has been lying around in the earth science community for quite some time. We've known that there are these kinds of fossil structures in basaltic glass on the seafloor, and they occur on the the outer crust of a basaltic pillow, if you wish, and it's just in that layer that we see those little spheroids that have been variably interpreted as fossil remains by Cavalazzi and colleagues. They've been reported from Cretaceous basaltic glass and even Paleo-Archean basaltic glass from the Huguenot Formation in South Africa, and they were interpreted in those cases as trace fossils. Hence, we call them ichnofossils. And here there's some detailed work that's been done by Kevin Lepeau and Karim Benzerara in France on these ones from the Neo-Archean of the Tumbiana Formation in Western Australia. And they found these kind of basaltic clasts, and they were filled with these titanite, and they reported these little yellow things at the end of those little microtubules to be organic matter. So the organic matter they claim was maybe derived by life, but the whole thing was not. So they concluded that these things are abiotic features. So you see, there's double explanation for these things. And I had the opportunity to visit a place where these were occurring in concretionary hyaloclastite. And I will show you a few pictures in the coming slides. Now I'm going to transport you to these very ancient hydrothermal vent environments in the Hudson Bay, right here in the Belcher Group, um, in the middle of the Superior Craton, which is color-coded in pink here. The age constraints are between about 2.025 and 1.870 billion years. The metamorphic grades are very low. The rocks are extremely well preserved. And it's in that formation, the Flaherty Formation, there's a number of pilolavas interpreted with pyroclastic flows and massive lavas. But there are layers that contain sedimentary rocks there. They're usually quite thin, less than a meter, maybe a meter, but they're quite thin. And in places, we can find them to be associated with hydrothermal vents. So I led this expedition back in 2011. We had an Inuit fishing boat and put a tent there. We had two little boats to go to shore, and a wonderful group of people with multidisciplinary backgrounds and some Inuit friends who were hunting while we were hammering rocks. Probably they were the more pragmatic ones. Yeah, so these are some pictures of the lavas of this Flaherty Formation. Beautiful pillows, some of them with this kind of weathering features, some large amygdules or vesicles inside. Some form massive columnar basalt, beautiful pillow structures, really. In some places, even pahoehoe type textures, which are indicative of subaerial deposition, whereas most of the pillow basalts are typically understood to form underwater. But right next to some of these samples, you see there were some of these concretionary features. So when I saw that in the field, I was like, that's something to sample. And it turns out that it was not really carbonate. The rock was kind of carbonate, but it turned out to be a hyaloclastite, which is kind of an explosive rock of carbonate matrix with basaltic glass shards in it. And so this was one of the key outcrops that I investigated. So there's a pillow basalt layer right here, and there's another pillow basalt right here. And then this interlayer had this kind of cloud of concretionary hyaloclastite, and it was occurring along strike with some Gaussian pods and some chimney of accretionary lapilli made of carbonate and silica. Really look like a white smoker type chimney here in this case. And these are ironstone pods of Gaussian and limonite, goethite, hematite, were probably some similar to black smoker, but these are geological evidence for fluid exhalations on the seafloor. And here are examples of some of the concretions that I sampled. And this is a wide range of rocks. Some of them mafic, some of them much more felsic. Most of them have carbonate or organic matter. So you have the numbers here, and I put them in this chart to show you the TOC content can be quite high, up to 5 or 8%. And the delta C13 value of the organic matter varies between -29 and -37 per mil. So that is quite significant and is not consistent immediately with oxygenic photosynthesis, probably more consistent with anaerobic heterotrophy or anoxygenic photosynthesis, possibly methanogenesis. The anaerobic heterotrophy might also be evidenced by these negatively fractionated carbonate carbon isotopes down to -11 per mil. And then the sulfur isotopes indicated quite enriched values indicative of sulfur disproportionation. But we could not necessarily rule out abiotic fractionation necessarily, but it's hard to conceive with such enrichments of organic carbon that Fischer-Tropsch type synthesis would lead to so much hydrocarbons being produced, especially at a time that we know was after the great oxidation event. So we know there was a lot of cyanobacteria around them. There was already a lot of life around then. So it's not a likely explanation, but we cannot entirely rule that out. So that's my cautionary word. So about this. But for the microanalytical work that we did with correlated microscopy approach, we looked at this thin section from this particular sample, And we could see the dark clumps here. These are all clumps of organic matter. So it's quite enriched in organic carbon. Here are two images in plane polarized light and cross polarized light showing the carbonate matrix and the basaltic glass shards, which are all kind of gray and isotropic with some amygdules in them. And some of these basaltic glass shards have these ichnofossils. So this is from a kind of a concretionary hyaloclastite, this concretionary hyaloclastite that has these objects inside. Some of them form little spheroids, a little bit like what was reported by Cavalazzi, and these microtubules, a bit similar to what was reported by Lepeau and Stogigel before. Even some of these tubes were seen here. These are kind of crossed uh, from the thin section process. They're kind of so they look like elongated structures, but they're really like cross sections of a tube that's inclined with respect to the plane of the thin section. And micro-Raman imaging showed that was some kind of phyllosilicate, was tentatively identified as chamosite, but definitely kerogen was there in red, and titanite was there, same composition as what had been documented by Cavalazzi, Stogigel, and Lepeau, and many others. So the kerogen is definitely occurring with the titanite, almost to the exclusion of the surrounding matrix of the basaltic glass shard. And ultimately, we did some focused ion beam nanofabrication, lifted out this lamella, saw the tiny, tiny size of this basaltic glass. And you can see here the small crystals of these phyllosilicates, which kind of have a regular type of composition dominated by silicon, aluminum. Some of them have a bit of magnesium and sodium, but wherever we see like on the side of the fib lamella, which was part in the tubule, part in the matrix, the side that was in the tubule, we could see this calcium and titanium composition that, of course, relate to the titanite that composes these objects. So the Minnesotite, pyrophyllite are tentatively identified as such based on these nanoscale analyses. In general, it's probably more like chamosite in composition. So we've looked at another one. Here's the first one I just showed was a microtubule. These ones are cocoidal ichnofossils, and they form these beautiful trails inside the basaltic glass shard. You can see the corresponding cross-polar image here. And here's a blown-up view of this part and a Raman hyperspectral image showing the titanite in purple and apatite co-occurring. That was very important because apatite is a very common biomineral. It occurs in our bodies, of course, in the form of bones and teeth. But also there was a kind of a line of red-colored organic matter that was a bit unexpected from these plane polarized light images, but still it was there. You can see the kerogen forming this line here in this hyperspectral image. Very strange kind of Raman spectra for the organic matter. Looks like a different kind of organic matter, maybe mixed with a bit of clays of chamosite, but nevertheless closely associated with apatite and also the regular kind of organic matter with these two peaks here, so-called the D band and G band. So we have a number of biosignatures here in the form of minerals, mineral associations, different kinds of microfossil-like shapes. We have uniformitarianism. We have the products of decayed life as apatite and as C13 depleted carbonate. So there's a lot of independent lines of evidence here. And I wanted to show this. This is just data I collected last week, so this is kind of fresh off the TEM. A fib lamella that was collected from right here, from this particular ichnofossil associated with apatite and organic matter. And this is what it looked like. There was an apatite crystal here. You can see it with what we call the poikilotopic inclusions of organic matter. These are tiny little inclusions. They're just a few nanometers in size, between 5 and 20 nanometers in size, and they are like grains of pepper inside this otherwise euhedral crystal of apatite. And we can see in the color-coded map here for the elements carbon and phosphorus just co-occurring beautifully on top of one another, just basically saying that this is from life. This is biomass, and it's inside the calcium titanium substance, which is titanite, and is an evidence that these are indeed um, uh, consistent with fossils or trace fossils. So to summarize, the evidence of life for this anaerobic for anaerobic life in the Flaherty is that we have C13 depleted graphitic carbon between -37 -29 per mil. We have a large range of C13 depleted carbonate, down to -11. Um, we have C13 depleted carbonate in concretionary structures, which is important because that's one of the product of CORs. There's a large range of delta 34S values in sulfides up to 12 per mil, consistent with biological sulfur disproportionation. We have tubular and coccoidal ichnofossils made of titanite, variably crystalline organic matter, and nanoscopic sized apatite crystals with organic matter, and the uniformitarian observation. So we have a number of independent lines of evidence that suggest that these life forms were probably anaerobic, but probably is always with a caveat. So the conclusions of my presentation are, first of all, we need to distinguish biosignatures from abiotic signatures. And so we need to consider the background definitions carefully. We need to consider absolutely the abiotic chemically oscillating reactions, which appear to be quite common in nature, as well as Fischer-Tropsch type synthesis, which also appears common in nature. Identify diagenetic spheroids if they are there. We need to consider the composition of the minerals. And lastly, I would say in general for biosignatures, we need several independent observations to make a case for early life. You cannot just rely on some microfossils and they look like they're made of carbon. You have to prove that they're made of carbon. You need to prove that the carbon has the characteristics that are expected of a rock that has been metamorphosed the way that the whole outcrop has been metamorphosed, at least. So we need information on the field geology, petrography, micropaleontology, geochemistry, and finally, I think we need absolutely uniformitarian observation. That's important to compare to younger rocks, older rocks, even modern rocks. Um, so these are my main take-home messages. So thank you for listening, and I'll be happy to take questions from here on.